G'day, I'm Heather Hussain and I'm a food scientist and I currently head up the Food Fraud Prevention Department at Food and Allergy Consulting and Testing Services in Stellenbosch. Today's presentation is about the role testing plays in validating and substantiating vegan labeling and claims. And I'm specifically going to focus on the challenges we've noticed and come across when it comes to the commercial test methods that are available, and how we've implemented a novel testing approach to help overcome these challenges. There's been an evident consumer shift towards plant-based products. So before we dive into the testing side of things, it's important to unpack what is meant by plant-based and how does vegan diets fit within this structure. Plant-based is the umbrella term, also sometimes referred to as plant-forward diets, for diet patterns that focus on foods primarily from plants. There are several variations within the plant-based group, often adding an extra layer of complexity for the food industry. By specifically having a look at vegan diets, you will notice that all animal and animal-derived ingredients are excluded from the diet. Legislation is the starting point to determine the requirements for product claims and declarations. So I will quickly touch on what is included and excluded in our regulations and why this is significant. Our current labeling regulations clearly defines vegan and how it should be indicated on a product label. However, it provides limited criteria on what is required for vegan products, especially in terms of manufacturing and testing practices. Therefore, the current regulatory framework do not support the industry in terms of addressing fraud, and the problem we've probably seen most is the cross-contamination from non-vegan sources. This has led to two major concerns. Although the allergic and vegan consumer are two different market groups, and allergen precautionary labeling is allowed on vegan products, we have especially noticed a confusion in industry regarding how to manage this, as 4 of the 8 common allergens are from animal origin. Since vegan indicates that there are no animal ingredients in the product, there is a serious risk to an allergic consumer who treats a vegan claim the same as an allergen-free from claim. In addition to this, neither the regulations or the guidelines accompanying the regulations address how to manage and control vegan products in shared facilities, and this is especially applicable to the South African industry. So why are we seeing a serious need for testing from the industry? Well, just to highlight some of the significant driving factors we have identified. Although testing to substantiate vegan claims are not mandatory in the regulations, industry is used to using testing as a tool to substantiate or validate labeling claims often. Multiple well-established and international vegan certifying and endorsing organizations include a testing component as part of their certification process in order to allow their vegan symbol or trademark on product labels. We have also noticed that the vegan consumer are not a homogeneous consumer group, meaning that their tolerance to the presence of animal compounds in their products due to cross-contamination differs. Many vegans have a zero tolerance, so testing is also driven by consumer assurance programs. Many major retailers in South Africa also request testing as part of their supply assurance programs. This all leads to a big demand for testing to ensure vegan product integrity. So we know industry wants to test, but what do they want to test for when they want to substantiate vegan labeling? Well, the objective of the testing will be to detect the intentional addition, so through adulteration, and unintentional addition, so through cross-contamination, for example, of a range of different animal ingredients and animal-derived ingredients, such as meat products, fish and shellfish, eggs, honey, animal rennets, royal jelly, and so forth. However, there's no test that does it all. Adding another obstacle to the mix is the limitations of the current commercial test methods that are available. From our observations, industry often turns to a combination of traditional species and allergen testing methods. Although these methods provide some information and a piece of the puzzle, it's not necessarily the most suitable method for the objective. DNA-based species testing techniques are frequently employed in isolation to confirm the absence of animal compounds in the vegan products. And there are several reasons why industry is using DNA testing methods for this. We have noticed that industry associates DNA with the presence of animal ingredients. However, it's not always a reliable target to identify animal and animal-derived materials. The commercial availability and accessibility also plays a role in the choice of analyses. And since these methods and techniques have multiplexing capabilities, meaning that several different animal species can be identified in one test, they are usually a more practical solution for the industry. However, there are many drawbacks for following this approach. DNA might not be detected for a variety of reasons, such as in the case of low DNA containing samples, example egg, Also, with highly processed sample matrices, the DNA might be rendered undetectable by the processing, and matrix influences and interferences can also happen with these techniques. In addition to this, there's also sometimes limited transparency in terms of the source of DNA that's detected. So is it coming from animal meat or is it coming from animal byproducts? A unique challenge that was also identified, and this is especially with South Africa, is the limited game meat species that are included in the multiplexes that are available. All these factors, regulatory limitations, the different tasting pressures, the complex nature of food, and the range of different animal ingredients, together with the challenges and drawbacks of the commercial taste methods, have left the industry with a problem in terms of how to go about tasting. For this reason, we have employed a novel tasting approach by making use of targeted proteomics by LCMS to overcome this problem and to support the industry. So what is proteomics? Well, in short, it's the study of proteins and their function, and it can be applied to determine the presence and quantity of particular proteins or peptides in a complex mixture of proteins. So how can we apply it to food testing? Well, since proteins are useful biomarkers in food analysis and are common analytes in a large range of animal and animal-derived ingredients and materials, they can provide evidence to confirm the authenticity of vegan products. Targeted proteomics offers a specific, sensitive, and accurate testing approach and is therefore a solution to the challenges faced with the traditional testing methods. But the proof is in the pudding. We have implemented commercial screens with the LCMS for vegan products and compared the results to our standard DNA testing. The DNA analysis were completed with a PCR microarray that qualitatively screens for the DNA of 24 different animal species. We have developed two different vegan screens, one for food and one specifically for wine to detect the different finding agents from animal origin often used in wine production. In the first case study, cattle DNA was detected in a feta and spinach pizza suitable for vegetarians. Although milk is a low DNA containing matrix, it is possible that the positive result was due to the feta present. Upon further investigation, the LCMS vegan screen indicated that both bovine muscle tissue and cow's milk was present, therefore indicating that there is cross-contamination from beef meat in the production facility, and this provided valuable insight to the industry. In the second case study, the aim was to determine the source of gelatin in a jelly sweet. The sweet also tested negative for any animal DNA. However, the vegan screen detected gelatin from pork. Since gelatin is a highly processed ingredient, the DNA was unable to effectively identify the pork species. Moving on to wine case studies, the first study was testing a positive control sample where pork collagen was used as a clarification agent in the production to determine if it could be detected. Once again, the DNA was not detected, but the LCMS vegan wine screen successfully detected the pork collagen. Collagen is a highly processed matrix and is often filtered out during the wine production, but the LCMS screen was sensitive enough to detect it and was therefore suitable for this purpose. Lastly, to also give a comparison of a traditional ELISA analysis method that was used for vegan wine tasting, which is often the case in industry, no milk proteins were detected in a positive control wine sample. However, the caseinates were detected with the vegan wine screen with the LCMS. This is due to the multiple drawbacks of ELISA, including not being sensitive enough in certain cases. Another interesting case study was when we detected chicken DNA in a vegan pasta, but after performing quantitative testing with LCMS, neither chicken or egg proteins were detected. So this left the question, was there cross-contamination with the chicken product or not, and was this contamination significant? In order to answer this question, you need to look at what the results are telling you. So looking at the DNA 24 species screen, you can note that DNA was detected. However, it's important to consider that a single strand of viable DNA can give a positive result, meaning if there's one strand of amplifiable DNA, you can get a positive result. And that means that one DNA strand does not necessarily indicate significant contamination. The results are also qualitative, and this is because DNA amounts vary within and between species, and the amount of target DNA might not be proportionate to the true amount of target compound present. Therefore, the level of chicken is unknown, and once again, you can't determine if there was significant cross-contamination. The method is also not transparent concerning the source of the DNA detected. So DNA from various parts or products of the stipulated 24 species can give a positive result, but the source of the chicken is therefore unknown. So you don't know whether it's coming from egg or chicken meat. And if it's coming from egg, it's an allergen, and that's more a serious concern. So it's really important to understand how you can interpret this data. Compared to the LCMS, it's important to note that LCM is quantitative, so it can tell you the level of contamination, and therefore you can have an indication if the contamination is significant. LCMS also detects species-specific peptides, so there's complete transparency with regarding what is being detected. Is it coming from chicken or is it coming from egg? So what do we learn from this? Well, once again, no test is the silver bullet, and result interpretation is dependent on the test method. And the suitability of the method will depend on the sample matrix and what you want to achieve by the analysis. And it's extremely important to understand this when using a test method. Therefore, in conclusion, there has been an evident consumer shift towards plant-based products. And this has highlighted the regulatory limitations that need to be addressed in order to have a regulatory framework that can effectively support the food industry. There is also several testing driving pressures at the moment, and combine this with the aspects of veganism that are unique and multifactorial, it presents a challenge when it comes to the commercial test methods that are available. Traditional DNA-based testing methods are often not a suitable approach in terms of validating and substantiating vegan product claims, and therefore novel testing approaches need to be explored and commercially implemented. Our studies have proven that targeted proteomics offers an accurate, specific, and sensitive solution to this problem. Thank you very much for allowing me to present this topic, and if there is any questions, you're welcome to let me know.